All right, we're going to start looking at macromolecules, so these large molecules, particular classes of these large molecules. And the one we're going to be starting off with is carbohydrates. So carbohydrates, again, commonly called carbs. As the name would suggest, these contain carbon and hydrogen. So their general formula is going to have one carbon for about every two hydrogens, and just generally speaking, there's also going to be some oxygens in there, and this, of course, is going to repeat. And so we're going to have several of these ratios of them, and it can vary depending on which carbohydrates you're talking about. The O's tend to be found in conjunction with the hydrogen, so we have these hydroxyl groups where you have this OH group hanging off of a carbon. And again, remember, when you have this OH group, you're going to have polarity because of that highly electronegative oxygen bonded to the hydrogen. You have the capacity for hydrogen bonding. They also, in terms of function for animals, is that they are, well, and plants as well, they number of different kingdoms, they are accessible energy storage, and so they're a way of taking types of energy and storing it as chemical energy. So a really nice way to store chemical energy in the form of bonds. They're built using monosaccharides. So these are mers, and if you take one of them, you call it a monomer, and mer is just a generic term. So if you're specifically talking about a sugar, you'd be talking about a saccharide. So one individual sugar would be a monosaccharide. That monomer can be bonded to another one, and another one, and another one, and another one, and you end up with a larger molecule, and we'd refer to those as carbohydrates. The link that holds these monosaccharides together is called a glycosidic linkage. So here's two common ones, alpha glucose and then beta glucose. And you'll notice that we've got this ring structure here. So there's one carbon there, 2, 3, 4, 5, 6. So we know the formula for glucose, C6H12O6. So that's the formula for the compound, and this is the structure with it. It has this hexagonal structure with oxygen being a component of that ring there. That makes it glucose. And then you'll notice that there are these little functional groups. And so there's the hydroxyl, there's the hydroxyl, and this one has the hydroxyl pointing upwards, this one has it pointing downwards, and then we have this carbon with another hydroxyl on it, which is actually able to rotate a little bit. And so the difference between a alpha glucose is you've got one hydroxyl down, 2 hydroxyl down, 3 hydroxyl up, 4 hydroxyl down. And when you look at a beta glucose, the first hydroxyl is actually up, pointing upwards instead, the way they are drawn here. Now the second one's still down, the third one's still up, and the fourth one's still down. And so the only difference between these two, between the alpha and the beta, is that on carbon number 1, the alpha has the hydroxyl pointing downward, and the beta has the hydroxyl pointing upwards. So these are isomers of each other, and that's the only difference between them. This does matter when they are used biologically to build larger compounds, because how that hydroxyl is oriented is going to determine how they come together, and then it's going to affect the shape of that overall carbohydrate. They can become linear, and so you may sometimes see them drawn in their linear structure, in which case the ring is opened up. And when that happens, they are able either to reform as a alpha, or it could reform as a beta, depending on where that hydroxyl group is pointed when the ring closes. So we have the option, the ability biologically for these isomers to convert from one to another, and there's actually enzymes that actually can convert from one type of isomer to another type of isomer. If you only attach 2 of these monosaccharides, so a large carbohydrate would be made up of many, you could call it a polymer, but if you only have one, you call it a monomer. And so I'm talking saccharides here, so a monosaccharide, but 2 of them together, it's a disaccharide. And you'll recognize the name of some of these. So if you take the monosaccharide glucose, and there's another type of monosaccharide, you notice it's got a slightly different structure here, same formula, another isomer, but built in a different way, and it's called fructose. So glucose and fructose, if you combine them together, We can do this through a dehydration synthesis where you have an OH and an H coming together and they form water. And then the carbon number 1 on the glucose here, and we have, what is this one? 1, 2, 3, 4, 5. Carbon number 5 on the fructose are going to end up bonded together through this oxygen here. So this bond here is called a glycosidic linkage, and it holds those two monomers together. The name of that particular disaccharide that's made up of a glucose and a fructose, the name is sucrose, sugar. Table sugar is commonly what people would call. And you can run this reaction in reverse. This is the same reaction above, but we're running it backwards. You can use water to split the glycosidic linkage. And so we call this hydrolysis. We're using water to split apart the disaccharide, and you get your glucose and your fructose back. With these isomers here, you can end up with different disaccharides. So in this one we have alpha glucose and alpha glucose. When they bond together, you get a disaccharide called maltose. And so you may have heard of that disaccharide before. It's not as common as sucrose, but it is another type of disaccharide. Quite confident you would have heard of lactose. Lactose is made from beta galactose and beta glucose. So again, isomers of glucose, but slightly different in their shape. And when they combine together, you're going to end up with this different type of bonding here, different orientation of these disaccharides, and therefore, different orientation of the monosaccharides to make the disaccharide, and therefore you get a different disaccharide. Something you may have noticed as we're going through that. So this alpha, and here we can see there's the beta glucose there. This tells us which isomer of glucose we're dealing with, but there's also in some ways, and you've seen on these diagrams, it may or may not mention it, you may see this other letter in there, D. What that is the type of optical isomer, and you'll notice they're all D's because the D optical isomer is the common one. Its mirror image, the L version of it, you're not really going to see in nature. All of our mechanisms for metabolizing and working with glucose are all based on using the D optical isomer. The L optical isomer wouldn't work. It'd be like trying to put your right hand into a left-handed glove. It wouldn't fit. And so what we see in nature is that D glucose and the D galactose and the D version of all these sugars here, that's the one that we happen to see. If you did have the L version of it, the mirror image of it, it wouldn't work with your machinery. So essentially you'd have a sugar that your body can't process. All right, so there's that in some more detail. And again, you'll notice if this were a mirror, you have a mirror image of the D version of glucose, and that's the common one that nature works with. There is the L version of glucose, no particular reason why it couldn't have been the L version, but it seems that evolution has favored the mechanisms to work with this one particular version that we now call the D version. All right, the glycosidic linkage that holds monomers together. So this one here, it's two bonds with an oxygen in the middle, and it's bonding those two monomers. There's two ways in which this can happen depending on, remember how when we had this diagram is a little bit more bent than what we were drawing earlier, but essentially we had, I'm going to take a shortcut and just draw a hexagon here. The OH could point up, and then the other option is you could have another isomer with the OH pointing down. And so depending on which isomer you're using, it can bond to another monomer in a different way. And so one of the ways we call an alpha linkage, and this is the one that is drawn here, and you'll notice that both of them have the bond sort of pointing downwards compared to where their ring is. So here's their hexagonal ring here, and they both point down from either side. So that's called an alpha linkage, and it tends to be bulky, and it ends up actually causing the monomers that if you were to do it again, so if they had another bond here, another alpha linkage here, and then you had your other, this isn't a great hexagon, but you can imagine that's it. What happens is it starts to spiral in on itself as it gets larger and larger as you add more and more of these monomers. That's an alpha linkage. Beta linkages, this is where you would have one of them pointing up and one of them pointing down in the particular way that they were. So let me just draw my hexagons again. So imagine, let's say I have this OH pointing up, and it bonded to an OH that was on another monomer that had it pointing down. So when they bonded, essentially they sort of like reposition themselves. So this one would have to go higher up. And let's look at the better drawing of it here. What they end up doing is they end up being quite linear in their arrangement. They're able to sort of line up sort of in a diagonal line to each other. And so it would actually bond up here, and you'd have your monomer here. And so you get this nice straight line when you have these beta linkages. So the takeaway is that alpha linkages are bulky and they tend to sort of curl in on themselves. Beta linkages are sort of off at an angle, and they can be quite long and linear and straight, flat structures. So because of this, we get two different large macromolecules when these linkages happen. With the bulky alpha linkages, we can make glycogen and starch. With the flat, sort of straight line way of doing it, we can get what's called cellulose. So these monosaccharides bonded together, again, you put many of them together, you get a polysaccharide. And the different types of these long chains of glucoses are shown here. So first one, glycogen. This is the one that our bodies produce when they're storing glucose. They can take alpha glucose and they can stick it together and they can build glycogen. It's a sort of a shorter term storage for energy. It's not so short term like glucose in your blood is really readily available. If you want to hang on to it for a little bit longer, say you drink a gallon of, I don't know, a slushy or something like that, it's got a whole lot of sugar in it, you're not going to use it all at once, so your body will store some of it as glycogen. It'll take some of those glucose molecules and it will start linking them together. And notice the type of linkage here. We have this downward circle downward, and remember what that does, it sort of relatively bends each unit next to the other one, so it's going to sort of curve over on itself and you end up with this bulkier structure. Starch, on the other hand, is again formed from alpha glucoses, but it is going to be for, we'll look at the picture in a second, it's actually less branched. And so plants use this one. It is still relatively short-term storage. So if you think of like a potato, right? It's got a lot of starch in it. That's how that plant is storing that energy for it. It is similar that we have these alpha linkages here. So it also is going to be sort of curled in on itself. And what's the difference between glycogen and starch is the branching. Glycogen is going to have lots of branches coming off of it. So we're going to draw a better picture in a second here. And starch has fewer branches coming off of it. Built the same way, just different in their number of branches. Cellulose, however, let's do this in a different color here, is made from beta glucoses. So it is going to end up with, and you'll notice this guy's kind of flipped over. This is probably an easy way to fit it into a box in terms of drawing, but not the best way to understand it. The way that these go is sort of one is relatively positioned to the other in a diagonal line. So I'll get a better picture up in a minute, but it forms these long straight chains. And as such, it's able to pack really closely together. So glycogen and starch made from alpha glucose, and they're bulky. Glycogen has lots of branches, starch has fewer branches. Cellulose is made with beta glucose, and it's going to form these long straight chains instead. So here is a starch chain of glucoses. So these are all alpha glucoses. So there it is there. And as mentioned, the difference between starch and glycogen is the number of branching. So starch can have some branching to it. Glycogen has lots of branching. And what it is you end up with these glucoses sort of lining up, but then every once in a while, off of the 6th carbon, you have this other chain starting, and so you end up with this branch forming. With beta glucose, you can make cellulose, and this is a better diagram to see why it forms straight lines. The other previous diagram was flipping every other beta glucose. This one has them all oriented in the same direction, and so just to draw attention to the linkage here, right? It's pointing up to the oxygen from this one monomer, and then it's bonded pointing to the next one as well. And so instead of having both of them pointing downward, causing a kink in the chain, these ones sort of line up in a nice diagonal line, and so they can form these nice straight lines that actually pack quite well together. This is the other way you'll see them often in textbooks. They will flip, so every other one has been flipped around. And you'll notice that because if you look at carbon number 6, it's on the bottom here, and then here it's on the top, here it's on the top, and then this one would have it on the bottom again. So they're rotating, alternating every single one. Same idea, a little bit harder to imagine in a three-dimensional, but the idea is it forms a straight line. So I would think of it diagonally or flipped going in a straight line. Either way, cellulose forms these straight line structures, not bent like when you have the alpha glucoses in starch or glycogen. So a little bit more on glycogen. Here's a larger view picture of it. So these little circles would be each one of them one of the monomers, and you'll notice that the purple ones outlined here is where you start seeing the branching. And so you have these branches coming off, and you'll notice that the chains themselves, because of those alpha linkages, they all start to sort of curl relative to the one beside them, and so you end up with what's a relatively bulky structure. Very well branched, and then the branches are between the 6th carbon of the running chain, and then it starts off a new branch with carbon number 1. It's about every 10 residues, which is another way of saying every 10 monomers, because they're not by themselves anymore. They're not really monomers, right? They're part of the chain now, and so we refer to them as residues. Starch is actually not, it's a pure substance, as it's a mixture, and so we have amylose and amylopectin. There can be some other things in there as well. So the amylopectin, anyways, very similar to our glycogen. It has that 1,6 linkage, so you're going along and you have these kinked alpha linkages. It's causing these chains to be bent, and then every once in a while, and it's not every 10 this time, it's about every 30, so it's about a third less branching, you have these branches coming off of them. Cellulose on the end, no branches. It is all 1,4 linkages, and again, here is a diagram where we have every alternating one's flipped, so it looks a little bit tricky. It looks like it's an alpha linkage, but it's because they've actually flipped every other one. So really they line up in a straight line, and you end up with the ability to pack them really, really tightly together. This is why you find cellulose in cell walls. It's a great structural material to make these fibers that you see in plants. If you've ever eaten some celery and you end up chewing on what's left for like 20 minutes, that's because of the cellulose, which is really well held together. It packs together in these really nicely tight packed fibers, make good structure for plants. Here's all 3 of them together. So again, starch, which if we look at one of the components of it, amylopectin, alpha linkages, branches every 30 residues. Glycogen, same setup, alpha linkages, bulkier structure, and it actually has more branching, so like every 10 residues-ish. And then cellulose, we've got no branching, and again, this is beta linkages. So you have one monomer, and it's joined to the other one in a beta linkage, which is then joined to the next one, and so you end up with this nice long straight chain, as opposed to these bent arrangements that you see with glycogen and amylopectin. Not as bulky, cellulose is not as bulky, and therefore it's better for building structures with, harder to break down. There is the final picture of all 3 of them. So again, we're using glucose to build all these, right? They're all built from glucose. The type of glucose that is being used is going to be different for cellulose. So we have beta glucose forming those beta linkages, and therefore you get long straight chains. If you have alpha glucose forming alpha linkages, you end up with the curving nature to it, and you also are able then to branch these more often. If it is glycogen, you're going to have branches every 10 residues, starch every 30 or so, and cellulose, no linkages, but packs really, really well together, good for structure. Starch, energy storage for plants, glycogen, energy storage for animals, and cellulose is not really used for energy storage, but instead used for structural support within plants.